thanks to Lubna, first of all, and you for giving such a nice introduction to stem cells. And what she was really focusing on, actually the clinical side of it, like actually the clinical trials that's going on. And really, people are actually injecting stem cells in the human body and hoping to cure the problems these patients are having, right? But my talk is actually as a molecular neuroscientist, like I am particularly focused on not on the clinical trials, not on the curing patients at this moment, future maybe, but right now we are really using the cells. What we can use the cells for is actually to understand the molecular mechanisms of proteins and genes that could not be studied before because of the reasons I'm going to just talk about. All right, so as this is my title, it is too long. Again, Jim changed my title. So I was changing, but then it's fine. We're going to talk about human neurons reprogrammed from model EPS and iPS cells, but also from cells like somatic cells like fibroblasts, etc., blood, etc.
So in my defense, let me just tell you that I had to send, when I wrote the grant proposal, I had to send in the titles of what the talks were going to be as part of the grant application, and I didn't want to deviate too far from what I said we were getting funded to talk about.
All right, so and that's the title of my talk, and you can see it's a beautiful neuron, and then people have been talking about this mouse neuron. People have been talking about dendritic spines and axon going on. Today, what I'm going to talk about how to make these cells from non-neuronal cells, make these neurons from non-neuronal cells. So we'll start on this slide about like how we go about like model neuropsychiatric disorders in the field for genetic mutations. We'll start on this slide about like how we go about like model neuropsychiatric disorders in the field for genetic mutations that actually happens to affect our cognitive ability and other psychiatric disorders, etc. So what happens is that we actually take the patients, we diagnose the problem, like they are having some sort of psychiatric problem, and we actually sequence them. For example, like a straightforward approach would be an exome sequencing, and actually we actually look at after sequencing we identify what kind of mutations that patient might be having. Like synaptic mutations, channel mutations, or genes in like the structural genes that might get altered, and these patients are having this sort of psychiatric disorders. So so far, like most of the time actually, we have been using different types of animal models to actually model those psychiatric disorders, and really to understand how these mutations are actually and dysfunction of the protein and lead to pathogenic phenotype. And finally, the goal is actually we actually build up on it. Is actually we go ahead and do the drug screening and also understand like what kind of side effects we might be having to make the drug even better. So, however, this approach actually has several issues. Actually, a lot of time making animal models actually is really a time-consuming process. Just making a knock-in animal, for example, takes or knockout animals. Takes years. I mean, those who are actually have generated animals, it takes forever. It's pretty expensive to breed these animals, like second generation, third generation. The scalability, how much you can keep, how many animals you can make. At the same time, a lot of times the genetic background is not really well preserved. The human and mouse and rodents, and a lot of times the genetic background is not well preserved in animal models. They are great tools, animal models. They are great tools, but not always. I'll talk about why. For example, like Noreen's talk also, when I asked her about, can you rescue a synaptogmin, loss of synaptogmin in flies by overexpressing human synaptogmin? The answer was no. There's something going on. There's a difference between synaptogmin in mouse and human that is actually is not exactly the same. So how can we study those genes? And also genes unique to human, that's not all expressed in mouse and very poorly expressed or not preserved well in mouse. How can we study those? And a lot of times we are also dealing with polygenetic disorders. What is polygenetic disorders? Polygenic disorders, a lot of time diseases are not happening because of mutation in a single gene, but multiple genes are getting mutated at the same time. And what you're really seeing is a convergent effect of all those mutations. So how can we make a polygenetic disorder? So how can we make a polygenetic disorder mouse model? You have to make single mutant animals, for that will take already a long time. Then you start breeding them, and then you second and third and fourth. You can spend your lifetime actually generating a mouse model for polygenic disorders. So now today I'm going to talk about an approach that can really change this long delay before we should get to understand the problems associated with polygenic disorders, etc., using a different model, that's the mRNA stem cell derived model. So just to let you know, to show you one example, just very briefly, that I'm not talking about about a hypothetical situation. I'm going to just briefly in 2 slides talk about one gene that our lab is heavily studying. Ours is a very new lab; it's been 6 months, but we are heavily studying a novel gene, a human-specific gene called the human neuroligin 4. Why is it very interesting in the context of cellular reprogramming in mRNA stem cell derived neurons? Because this neuroligin 4 is a synaptic cell adhesion molecule. This nucleus, and it's a part. It's one of the members of neuroligin family. In the family, there's like 4 neuroligin genes: neuroligin 1, 2, 3, and 4. And what you can see, this H stands for human, and M stands for mouse. So what you can see, the other family members, that neuroligin 1, mouse and human, neuroligin 2, human and mouse, neuroligin 3, human and mouse, they're very similar to each other in terms of their amino acid sequences. However, when we come to neuroligin 4, a gene that is like heavily mutated in autism patients, the human neuroligin 4 and mouse neuroligin 4 has 51% sequence similarity. So it's a very different gene. On top, actually, go and the cortex and run a qPCR for expression of neuroligin 1, 2, 3, and 4. And what they found actually, that compared to neuroligin 1, 2, and 3, neuroligin 4 in mouse, this is data from mRNA data from mouse cortex, is really low. So the expression level not only is low, poorly conserved, but also the expression level is very low in mouse. But as I mentioned just a second ago, that it is a massive number of mutations. One of the mutations we are studying right now in the lab. But there's many mutations in this gene that are directly associated with autism patients. Sources in the Simon Foundation, Autism Research. And as I mentioned, of course, like because it is highly disease relevant, disease in autism has been found to be mutated. Of course, we want to know like how it is expressed in human brain. So we actually, me and Sam, we actually got. And as I mentioned, of course, like because it is highly disease relevant, disease in autism has been found to be mutated. Of course, we want to know like how it is expressed in human brain. So we actually, me and Sam, we actually got collaborated with a physician in Utah. And then we actually got samples of human brain samples. These are postmortem brains, and then we got samples from different areas of the brain actually. And we run a qPCR to see how the expression compared to GAP-DH. And we see like roughly around 2 to 4%, 2 to 3% or 4% expression in neuroligin 4 in various tissues of neuroligin 4 expression. Not only the mRNA level, but when also we actually did a Western blot to verify the expression. And we saw particularly in frontal temporal lobe, parietal lobe, these are actually cortical lobes, is very high expression of neuroligin 4. And so we can actually draw a map that like in the cortical area is neuroligin 4 is very high. So I'm not going to talk about too much about neuroligin 4. I'll come back to it later on. But I'm just trying to give you an example of how can we study a gene that is poorly conserved, poorly expressed in mouse models, which is very highly expressed in human brain and associated with disease. I'm going to talk about now the alternative route, how we can do that. So the alternative route is called cellular reprogramming, and some of you may have like just seen this couple of slides, next few slides that people talks here in the BMD. So I always talk about self-headed identity, where actually Lubna just talked about that, where we can all our actually the pluripotent cells actually give rise to all kind of terminally differentiated cells in our body. And then, for example, she was talking about really mRNA stem cells can give rise to any kind of cells like fibroblasts, blood, and etc., etc. However, the iPS cell reprogramming, the where it's a process, what Shinyamana's group in 2006, again, and etc. etc. However, the iPS cell reprogramming, where it's a process, what Shinyamana's group in 2006, again, she briefly touched upon what they showed. Actually, you can take a terminal differential cells like fibroblasts, blood, etc., and you can overexpress transcription factors like OCT4, SOC2, FRET4, CMYC, etc. And when you overexpress these transcription factors, you can de-differentiate. So this process is called differentiation, and now you go up the hill. This process is called de-differentiation. So you can de-differentiate the terminal differential cells and actually bring them back to a state where they kind of mimic the stage of embryonic stem cell. This stage is called induced pluripotent stem cells. Induced because you're inducing this pluripotency in stem cells and you bring them into a condition where you can differentiate these cells into, again, any type of cells you want. You can make an alternative fate, get an alternative fate. For example, our differentiation protocols to make neurons out of it very efficiently. However, this whole process that why it takes this whole process of actually from getting a terminal differential cells to make induced pluripotent stem cells, the whole process takes pretty long time. It takes years, and sometimes you have to make sure that it's clean and it can generate. It is actually stem cell. It is actually capable of differentiating into 3 different germ layers like.
Just talking to myself.
Okay, sorry. It can generate mesoderm, ectoderm, endoderm, and it has the ability to actually differentiate to different types. So the whole process takes a long period of time to generate iPS cells, although this process is rather straightforward. So to avoid this year-long wait in this patient, recently, actually, our lab, both of them, my 20s group and Harry Winthrop, certain types of transcription factors. These are cell type specific transcription factors, and you can directly convert them into another type of terminal differential cells. You don't have to go to the iPS cells and come down the hill, and that this saves you a lot of time because this process, as I mentioned, takes the most time, like that, about a year or so. So this way you can actually overexpress lineage-specific transcription factors like MyoD. When you overexpress in fibroblasts, MyoD expression gives rise to muscle cells. You can express brain 2, ACL1, nephron-like. You can also convert fibroblasts directly into neurons. And this whole process, actually, at least in mouse, initially was shown in 2010 when I was finishing up my PhD. I was like final year, and then it really caught my attention that transcription factor 3 weeks, only 21 days, they completely changed the morphology. These BAM factors we call it, they trigger, and then it really caught my attention that transcription factor 3 weeks, only 21 days, they completely changed the morphology. These BAM factors we call it, they 2 ACL1 and just to give it a cool name. And they completely changed the morphology. And not only they actually have the beautiful, they have beautiful this neurotic processes, neural outgrowth, morphological changes, and these processes, as I mentioned, it takes only 21 days, only 3 weeks, and it's a very homogeneous population. All these neurons detached from, they're more or less like very similar. They have all the functional properties, and functional properties I'll talk about later, but at least morphologically they look very homogeneous. And they have extensive neurite outgrowth, as you can tell already from this data. At the same time, the question was also like if these neurons express neuronal mark in these cells actually generated from reprogramming, do express neural markers, how to measure them using an electrophysiology rig, and how to measure ion channels, particularly these two for today. But then neurons do actually have these properties, and it's very important to see actually these cells are completely functional. At the same time, neurons don't live in isolation. They should talk to each other. They make these synaptic connections with each other where they actually exchange electrical information. So does these induced neurons have these functional properties? So for that, and also the marker expression. So the first is the marker expression. As you can see, these neurons actually express TUJ1. It's a beta-3 tubulin expression. It's specific to neuron. So it converges TUJ1 expression. And then tau is another, Jim was talking about tau GFP. expression is there. Then TUJ1 and MAP2 and a new one is actually mature neural marker, and that expression is also there. So this, all these pan neural markers are there. At the same time, they have really beautiful like punctuated distribution of this, synaptic markers like synapsin. This is actually really, the synapses are formed along the dendrites, like in specific locations, and you can actually really label them with the synapsin markers, like as a punctuated distribution that has a punctuated distribution. So yes, so neural markers, these cells actually express TG1 or MAP2 and NUN, etc. At the same time, they also have synaptic proteins like synapsin. So what about the functional properties of these neurons? For that, again, I'm going to show you guys like how to do this process. Basically, we actually make attachment recordings from a single cell, and as you can see, I actually have other cells around it, but I'm catching these particular cells, and the catch pipette has an Alexa dye, a fluorophore, and that can actually load the cell with the dye, and you can really visualize what cell I'm catching. And you can depolarize the cell, you can hold the cell at a particular voltage. Sorry, this is a current line experiment, I guess. It is a current line experiment. So basically, you are not injecting any current first, and then you are injecting small little currents in it just with a square pulse, and you can actually depolarize the cells, and the cells can fire beautiful action potentials, right? And then at the same time, you can do also voltage line experiment, where you actually hold the cell at a particular voltage. For example, here I'm holding the cell at minus 70 millivolts, and actually stepping up, in holding it at different voltages for a brief period of time. And what you can see a beautiful inward sodium current. That's always, there's a sodium influx, and you can see outflux of the outward potassium current. And you can also magnify this little area, and you can see beautiful inward sodium current inside the cell. Okay. At the same time, as I mentioned before, the cells are also make synaptic contacts with each other. We can measure them. So when we record from them, we see these beautiful synaptic events. These are actually single vesicles releasing from the presynaptic terminal, and we can do voltage current recordings to measure them. These are called miniature postsynaptic currents. At the same time, you can also stimulate the presynaptic nerve terminal and cause all of these events to release at the same time, giving you a gigantic response. That's called evoked postsynaptic currents. Okay, so yes, these cells actually not only they look like a neuron, they act like a neuron too. So they have the intrinsic properties and they have the functional properties as well. Over the period of time, like our group and many other groups actually have shown that it is possible to use different types of transcription factors to generate different types of neurons from different sources, like you can use mouse fibroblasts, hepatocytes, or human sources as well, and you can make types of neurons you want. What kind of transcription factors you choose here?
You have no supporting cells in the culture.
Oh yeah, I did not talk about it. You have glia. Yes. We do have, we co-culture them with glia. We co-culture them with glia, mouse glia. You can also make human glia. There are protocols out there too. You can use SOX2 and IgF. I forgot the factor. There's two transcription factors.
You get a different phenotype depending on what glia.
They actually did not. The paper just came out about, it's one of my colleagues actually, he got a position at Lund. The paper came out in Nature Methods about like 2 months ago. Actually, I asked him. They did not compare side by side, but that will be an obvious thing to test actually if there's a difference. What kind? Thanks for giving me the break. I need a water. Yeah, so we can make different types of neurons. Yeah, so we can make different types of neurons, and depending on the transcription factor we choose. But then you can think about like skin, and also I was talking about hepatocytes. Who's gonna get like lung tissue, or like even like skin is a little bit invasive. Like pricking people like getting tissue samples, skin samples is kind of painful, and it also takes a long time to replenish. And also it is just difficult to get. So we were actually wondering if there's other source getting these cells, like somatic cells, and it can be also converted. The easiest is actually to collect from human body is actually from the human blood, because you get a ton of it. I don't know how much you get, but a lot of. Yeah, I don't know actually how much they collect in fact, but it is definitely less invasive. And then it is right here, neurons directly. So what we did actually, yes, if you use the same type of transcription factors, it takes a little longer than fibroblasts, but then you want after 2 weeks, after 3 weeks, and after 6 weeks, actually there you can see their cells are completely, these are blood cells with T lymphocytes, they're completely changing, they're changing their morphology, and they fire beautiful action potentials. And at the same time, you can get, when you co-culture them with mouse glia, as brought up, Mike brought up, when you co-culture them with mouse glia, you can get beautiful network activity. At the same time, you get spontaneous postsynaptic currents, and you can block them with blockers, synaptic receptor blockers like CMQX, AMPA receptor specific blocker, toxin GABA A receptor blocker. When you put them together, you completely get silence. At the same time, not only the spontaneous postsynaptic currents, you also get a beautiful evoked postsynaptic currents with beautiful delayed release. You also get a beautiful evoked postsynaptic currents with beautiful delayed release component, asynchronous release component. And again, when you add these drugs, so yeah, you can take like blood, like T lymphocytes, T positive lymphocytes, you can make functional neurons out of it. It takes only 6 weeks. Yeah, but then there's a limitation of the somatic cells. The limitation of these fibroblasts or blood cells, the problem is actually and make them neurons is actually you have to, if you want to study a particular gene, you have to make a knockout or knockdown of a particular gene. So, but the G cells are actually to generate particular introduce a specific mutation in it. It is very hard to do gene targeting in the cells. At the same time, is also the availability of tissue samples. No matter how much you get, at some point you will run out of it, right? And at the same time, you also, if you want to do structure function analysis of a gene of your interest or protein of your interest, you cannot really use the cells because you have to wait for someone who doesn't have a gene of your interest as a knockout for that gene, right? Or a mutation that you want to study. So what you can do, you can actually use ES and iPS cells, and stem cells or iPS cells to circumvent that problem. Because this ES and iPS cells, as Lubna again mentioned, not this part, but the self-renewal capability that like ES and iPS cells will actually grow infinite time. So you will never run out of your source. At the same time, gene targeting in embryonic stem cells and iPS cells are a lot more easier. So for that, and so then it'll be easy to make neurons or other types of cells when you have that source. This one.
What about siRNA?
So we did extensive amount of siRNA knockdown. So for that, actually, we were looking for ES and iPS cells, effective transcription factors that can actually convert ES and iPS cells into neurons. But not only transcription factors, I'll briefly also mention other protocols like small molecules we can also use to actually change embryonic stem cells into neurons. Actually, you did some of it yesterday. Whoever did the practicum with Lubna, you guys used some cells that were treated with some small molecules to make NSCs, neural stem cells. So those cells, I don't know. So how to generate human neurons from ES-iPS cells? For that, we use a transcription factor called neurogenin-2. A lot of time we use this transcription factor because this transcription factor is highly expressed as a chrominal transcription factors, highly expressed in the dorsal telencephalon during cortical neurogenesis, and we actually put them into a doxycycline-inducible telencephalon during cortical neurogenesis, and we actually put them into a doxycycline-inducible promoter with a selection cassette for pyromycin resistance cassette, and so we can select for the cells that were infected with NGN2. And we can actually see within -2, like where the cells are still ES cells, and as soon as we overexpress neurogenin-2 in the cells, within 4 days, actually, they completely convert, change their morphology, and they look like they have a small big process. look normal. At the same time, the mRNA stem cell markers, for example, NANOC is a strong mRNA stem cell marker that actually completely disappears in cells within 4 days of transgene expression and Gen2 expression. And they have this GFP to actually see which cells are these, and these cells are completely staining negative for NANOC. Within 4 days, they completely shut down the pluripotent. They have a rapid loss of pluripotent markers within 4 days and rapid neurite outgrowth. So we actually wanted to know how well these cells actually recapitulate the normal neuronal developmental processes. So what we did, we actually co-cultured. This time I have it on the slide. We co-cultured them with mouse glia, and we actually looked at the cells at day 7, day 14, 21, 30, and 45 days. We actually did an enormous amount of imaging analysis for marker expression. At the same time, we also did an enormous amount of electrophysiological analysis to understand how these neurons develop over the time in culture, in vitro. And does that really recapitulate at least some of the in vivo neurodevelopmental processes? So first, let's look at the markers associated with the early neurogenesis. For example, TUJ1 is actually a neuronal marker that's very highly expressed in early. When the neurons are born, they are already very highly expressed in TUJ1. And then the SOX2 is actually an mRNA stem cell marker. At the same time, nestin is actually an PC-NSC marker. So this is actually still the cells are sitting at the mRNA stem cell stage, where you're seeing them with dumping is a cell actually dividing. They're this plane, and they will be pulled away from each other. And then they're staining brightly with SOX2, but they're staining mostly negative, just nothing with TUJ1 or nestin expression. So what happens at day 4? Day 4, already you see SOX2 expression is completely gone. So the mRNA stem cell markers, pluripotent markers, completely gone by day 4 after transcription factor induction. And they start expressing TUJ1, early neurogenesis marker. And they also briefly overexpress also the nestin, that is the NSC marker. At day 7, what happens is again, they always stay, day 7, they always stay negative. So this is the mouse glia. We are co-culturing with the mouse glia. But then if you look at the human cells are actually indicated with white arrowhead. Those are always negative for SOX9, for SOX2. And the nestin is actually upregulated during NSC because this neural stem cell marker, and then again it kind of downregulated. And they stay like that for the entire time. We actually image them during the entire time. Okay, so they have a robust expression of TUJ1. They have transient expression of nestin, and then it goes down, and they have zero expression from the beginning. As soon as you make neurons, SOX2 expression is completely gone in the cells. Okay. The same way, double cortin is another marker, DCX. Double cortin is another immature neuronal marker. These are markers, actually, I should have mentioned. These are the markers people doing in vivo studies in mouse or other organisms always use these markers to identify what kind of neurons it is, what stage they are in vivo system. We are seeing this exactly same thing in vitro system too. So they actually do immediately upregulate the double cortin, that is actually a mature neural marker, but at the same time, and also the TUJ1 is always there. Whereas Ki67, which is actually a proliferation marker, Ki67 is high in mRNA stem cells. It's obvious because mRNA stem cells divide very fast, right? So it's a highly proliferating cells stage, but then as soon as you make neurons out of it, they are never dividing. They're always completely shutting down the Ki67. It's completely black. At the same time, what about mature neural marker like MAP2? Is a microtubule associated protein, is expressed only when the neurons are very, very mature, functionally mature, sometimes also mature neural marker. Again, in vivo, people who are doing in vivo slice studies are always using these markers, these mature neural markers to identify mature neurons. You can see UN, on the other hand, sort of gradually develops in the cells. So these cells are already becoming neuron immediately within 4 days, but gradually becoming more and more mature in terms of their marker expression, UN and MAP2 at least. At the same time, what about synapse? You have a question. Looks like you have a question.
Yeah.
So these images suggest that neurons aren't always growing on top of the glia.
These neurons suggest that no. So these are monolayers.
Yeah.
Most of the time. So they are surrounded by glia, not always growing on top of the glia. So we limit the. Yeah, so I try to actually decrease the amount differentiate between human neurons from the glia, so because there's too much signals. Of course, you can make it high density cultures. Even people, I will be showing you, people are making organoids. They are like dense, packed with cells. So I'm just showing you just the monolayer protocol so far, that where you can still see some sort of like gradual development of the neurons in terms of maturation, in terms of their expression, marker expression, also the functional maturation as well.
Do they get myelinated?
Huh?
Do they get myelinated, these
They
accents?
definitely get myelinated. Otherwise, you see the action potential firing weird. They will
But
not
for
be-
that, you need to co-culture with other cells.
I will show you. I'll show you guys. Not myelination part. People have done myelination studies in these cells also. The paper is still not published yet. But at least these cells are firing beautiful action potential, so it has to be myelinated. Otherwise, it will get diffused away. Sodium. Have done myelination studies in these cells also. The paper is still not published yet. But at least these cells are firing beautiful action potential, so it has to be myelinated. Otherwise, it will get diffused away. Sodium. So at the same time, synapsin is another really mature neural marker. What you can see from MAP2, the synapsin protein is also getting punctated distribution, is getting up and up, and the expression level is going up and up. So reprogrammed neurons, actually, these human neurons actually gradually express mature neural markers as well. So this slide will be a bit crowded, but on purpose to give you a full picture of all the marker expression in these cells, and hopefully I'll be able to convince you that these cells, the kinetics of the cells look similar to what we have been seeing in the development. So as I mentioned, to summarize all the data I've shown you, these cells immediately after your transgene expression immediately shed the pluripotency markers and proliferation markers like nano, SOX2 nano is not copied here, but then SOX2 and Cas7 within 4 days, you completely shut down these pluripotency markers. Transiently upregulate these NPC markers, the neural stem cell markers, and then downregulate slightly also after that stage. But at the same time, they rapidly acquire neuronal markers like TUJ1, double cortin that is supposed to be expressed in the immature stage. However, when it comes to mature neural marker like MAP2, NeuN, synapsin, MAP2, NeuN, and synapsin, you see the same trend. They're gradually getting up and up and up. So they're becoming neuron very fast within 4 days, but they're slowly becoming more and more mature over time. What about the kinetics of functional properties? That is also a very, very important. You guys can explain it. So what I did is actually made recordings at all those developmental time points, day 4, day 7, 14, all the way to day 45. As you can see, the cells are actually firing more and more, and you can actually quantify that. The cells are already at day 7. All the cells are firing action potentials. Although the number of action potentials is low, but then this suggests at day 7, near 100% of cells are firing action potential. That clearly suggests the cells are becoming neuron very, very fast with this transcription. However, they continue to actually mature more and more during this time point, over the time. Not only the action potential firing, another thing is like I'm a synaptic biologist. I'm very much interested in the synaptic plasticity and all. That was my graduate school theory, so have to do some synaptic recordings. So when I again made recordings from them, we actually made AMPA receptor mediated evoked EPSCs, and this is the stimulus artifact when you stimulate a cell. And this is the stimulus artifact when you stimulate a cell, presynaptic terminal. You generate this artifact, this electrical transients that directly come from the stimulator, stimulating electrode to your recording electrode, but also followed by this beautiful inward current that is actually driven by a little slower because you are stimulating the presynaptic terminal this time. You're releasing all the content that is being received by postsynaptic receptors, opening up and then allowing the sodium ions to actually flow in, and that's giving you beautiful inward current. These are called excitatory postsynaptic current or EPSCs. So given the time period of development of time frame, what you can see day 4, day 7, 14, 21, and the synaptic currents are getting bigger and bigger and bigger. And it's quantified over here that this neurons, NGN2, neurogenic 2, when you overexpress the cells, you're actually making like 100% of, not yet. That's the next click, not yet. That's the next click. These cells are actually at least gradually maturing, getting more. Sorry, I just made the slides this morning. Getting more and more mature. Interestingly, when you add drugs like CPP, which is an NMDA receptor blocker, and CNQX, which is an AMPA receptor blocker, these responses get completely blocked. So there will be no responses. All the responses in day 45, they will look like this. So there's no EPSCs anymore, suggesting these type of NGN2 dependent neurons derived from NGN2 cells are actually glutamatergic neurons. So they do not have any other type of neurotransmitter. So they're completely shut down if you add CNQX and cell. How about, can we also make, we made glutamatergic neurons, can we also make GABAergic neurons using a different transcription factor? So in the eminence area, so as opposed to glutamatergic neurons, cortical glutamatergic neurons, the GABAergic neurons in the cortex are not generated in the cortex. They are generated in the ganglionic eminence, MGE, LGE area. They're generated there, and they sort of like migrate they go to different places, including cortex. They go to striatum, they go to olfactory bulb, they go to cortex. So they are generated in different places. Also, they express different types of transcription factors. So we screened for that, and we came up. I'm not going to show you the whole thing, but the summary, the transcription factors that work best for us, there is AACL1, and at the same time included at DLX2. These genes are very highly associated with GABAergic neurogenesis in vivo. So we pick those transcription factors, put them under a tetracycline inducible promoter, and overexpress them in embryonic stem cells. And this is what we get. We get AD cells, ACL1, AS2, we put together AD cells. These cells actually, as opposed to NGN2 cells, AD cells, these cells actually, as opposed to NGN2 cells, NGN2 cells actually do have expression of D-GLUT1, that's the glutamate transporter that is associated with the excitatory neurons. These cells do not have any D-GLUT1 expression. Sorry, any D-GLUT1 expression. As opposed to that, they express VGAT, GAT61, and GAT65, GAT67, and GLX gene, GAT genes. So these are actually inhibitory neuronal marker. So these cells, AD cells, as opposed to NGN2 cells, but NGN2 cells, on the other hand, is negative for all those GABAergic neuron markers, suggesting AD neurons, as opposed to NGN2 neurons, make only inhibitory neurons. So we also, I wanted to know what kind of inhibitory neurons they are, because there are many types of inhibitory neurons in our brain. Some are somatostatin positive, some are somatostatin positive, some are PD, paradopamine positive, calvinin positive. We found out that, like, from our qPCR data, actually this was RNC data, bulk RNC data, what we saw that these cells are actually a mixture of cells that are actually expressing calvinin, somatostatin, VIP, NOS, NPY, etc. So these are actually these GABAergic neural markers that are present. Again, people actively use the in vivo. People actively use these markers to identify type of GABAergic neurons expressed present in our cortex, in human cortex or mouse cortex. And how do they look? Are they all expressing all markers? No. Some of the cells are actually, all the neurons are expressing POXU1. POXU is a 4-vein identity. It actually tells you that these neurons we are generating have a 4-vein identity. At the same time, they're also expressing GABA, so all neurons are GABA. And they are also expressing GLX genes that we are expressing, and also they have GAT65, GAT67. But the calvinin, somatostatin, and PV is actually subsets of cells that are actually expressing those individual markers, suggesting we are making, with this protocol, a mixture of different types of GABAergic neurons that are present in the human brain, in the human forebrain, in the cortical layers. So what about the functional properties? Do we see also not only GABA expression, but do we also see it in the synapses? Do we see GABAergic synaptic transmission as well? For that, again, we do electrophysiological recordings, and we see people who are physiologists like Mike and Matt, and you guys already know that this slowly decaying current with slow decay is actually represents a GABAergic synaptic transmission. And not only spontaneous, but also we can stimulate the presynaptic nerve terminal and actually cause the release of the GABA from the presynaptic terminal, and we can record it from the postsynaptic cell, not only in single stimulation, but we can also do trains, and we get beautiful this spontaneous, this postsynaptic, this delayed release, asynchronous release component that is coming after the stimulation. So these are beautiful GABAergic synapses. But why would you take my words for granted? So when you add picker toxin, you can completely get rid of all the responses from the cells. So these cells are purely GABAergic neurons. So now we have protocols, transcription factor dependent protocols that can make with NJ2 that can make glutaminergic neurons. Now you also have neurons that you can make GABAergic neurons that you can make with ACL1 and DS2, and you can mix them together to generate something like this, where the cells are labeled red. These AD cells, these inhibitor neurons, are labeled red with the primary neuronal culture, primary mouse neuronal culture. You have beautiful network activity. If you spikes, these are network currents, like where they're recurring, they're talking to each other in cycles, so the network activity. At the same time, if you magnify the small little area, you see this fast GABAergic, glutaminergic synaptic transmission, which is labeled with this green asterisk. At the same time, you also get slow decay events, which is slow decay that is actually GABAergic synaptic transmission. So you have both EPSCs, marked in green, that's excitatory postsynaptic current coming from the glutaminergic neurons. At the same time, you have IPSCs, that is inhibitory postsynaptic current coming from GABAergic neurons. And this time, you have to add NQX, CPP, and picker toxin, all three of them, to get rid of all the currents, suggesting you have a mixture of different types of events. You have a mixture of different types of events. And as I said, like in mouse neuronal culture, if you take a primary neuronal culture, as described by previous people and giving talks, and you see exactly the same. So now we have a way to make exactly the same mouse human primary neuronal culture-ish condition, where you can test many things you want. So yes, synaptic integration, excitatory and inhibitory neurons, they actually talk to each other, start talking to each other when you put them together. So I will briefly touch upon this thing. I'm mainly focusing on transcription factor mediated reprogramming, but it is also possible to make neurons without transcription factors by blocking different pathways. For example, if you block using different small molecules, for example, if you block dual animation using BMP, block the BMP pathway and TGF-beta pathway using small molecules. You can first make NSCs, neural stem cell beta pathway, using small molecules. You can first make NSCs, neural stem cell-like cells, and then from that you can make neural cells. What do I mean by that? So you have mRNA stem cells, as I mentioned before, they're highly expressed in animal. SOX1 is not expressed in neurons, but it is expressed in NPCs and NSCs. Neural stem cells. So, and TUJ1 is only expressed in neurons. So, when we, after a couple of days, when we use these cells into NSCs, neural stem cells, you can see high expression of SOX1. So, we're making from mRNA stem cells, from mRNA stem cell stage, when they had zero SOX1 expression. Now, by day zero, in 6 days, you can make cells that has high expression of SOX1. And the TUJ1 is still not there yet, but it will come soon. By the way, these are the cells actually you were imaging yesterday using alkaline phosphatase, whoever went to Lukeman. And at the same time, after 5 days, from day 0 to day 5 days, you can give it another 5 days, actually plate them, take them off, dissociate them, again plate them. You start after 5 days and provide some other small molecules, and I'm going to go into that. you can start seeing expression of TJ1, that is a neuronal marker. So in this way, you are making first from mRNA stem cells to neural stem cells, from neural stem cells to neurons. And at the same time, again, using another marker like OP34, so this stays positive for mRNA stem cell that completely disappears at this stage. Double coating marker, neuronal marker that I mentioned before that starts expressing. And PAC6 also, you see, PAC6 is an NSC marker that got expressions also using dual SMAC initially. And if you put all the markers put together, all the markers that I just showed you in the previous slides, if you put all the markers together, you see these are actually cells, NSCs, non-stem cells, that are expressing SOX1, as I mentioned, PAC6, and JAPI, they look like this, GNF, GNF, and G3, etc. Then you get as beautiful looking NAV2, JAPI, GABA, some cells are GABA, so this is actually a mixture of cells. Are these chemically induced neurons are also functional for that? We actually, not only morphologically, but also functional properties for that. So, yeah, this is how they look after a couple of days, like 12 days, 14 days, TJ1 expression and JAPI expression. And we can definitely record from them, physiological recording from them. And yes, they have beautiful action firing, they have inward sodium current, outward potassium currents, etc. So they are fully, these chemical induced neurons are also, this is all neuronal markers, they are also exhibit functional properties of mature neurons. Okay, so so far I've been like showing you all these generation of neurons of different ways. Am I running out of time? I am. When am I supposed to stop?
Noon.
Oh, noon. Oh, neurons of different ways. Am I running out of time? I am. When am I supposed to stop?
Noon.
Oh, noon. Oh, then I will just skip everything. And I would say this slide, where I would say that like this is the slide I just started with. I was saying that like animal model problems, and then right now hopefully I was able to convince you that instead of animal models, now we have the means to actually make neurons, actually human neurons of human origin, using different approaches where directly reprogram, we can actually make fibroblasts or blood cells and directly reprogram them into neurons. And when scalability is a factor, when we need more of it, then we can actually make iPS cells and then have an iPS cell bank and then actually differentiate them into neurons and study all kind of stuff that we want to study. Understand molecular mechanisms of genes, at the same time understand pathogenic mechanisms of different genes, different mutations in different genes. At the same time, we can also show like these cells are actually, we can use these cells for drug screening. Again, I think that's time to talk about. With that, the last point, not only these monolayers, we can make beautiful organoids, cellular organoids, which is a three-dimensional construct where cells are actually folding and actually making these layers where that happens during cortical development, layering of different types of neurons. At the same time, you can also transplant these cells in mouse brain, and then also they migrate into different areas. So it's a very fun time, an exciting time to be in stem cell biology, not only the transplantation side, the clinical side, but to understand molecular function using a human system, a human neural environment. So none of the different progress actually using the system in our new lab. These are the new lab members: Lisa, Tom, Lisa, Tom, Daniel, and Harper. We're actively working using these protocols to understand neurodevelopmental function, the mechanism, etc. And I'm affiliated with. I definitely have to thank all my past collaborators at Stanford, and many of them, and also San Buffalo, many of them actually have gone out now. They have their own positions, and we are talking, we're collaborating too. Thank you for listening.
So what about the use of these skills for neurogenetic disorders?
Yeah, so
From a patient?
Depends on what kind of degenerative disorders you're trying to. Of course, people are using these cells to understand. For example, ApoE, for example, is actually causative for ApoE4 version is actually associated with Alzheimer's. So people are using how the mechanism of ApoE4 versus ApoE3 versus 2 versus 1, how these different versions of ApoE can have people are using it extensively for neurodegenerative. But for our lab, we are mostly focused on the psychiatric.
Okay, so for example, if you would start with fibroblasts from young adults versus an aged person, you would recapitulate aging in neurons?
Yeah, so usually what happens when you make iPS cells from somatic cells, you kind of like erase most of the epigenetic markers, a lot of it actually, not all, but a lot of it. So direct reprogramming, on the other hand, retains a lot of it. So using direct reprogramming in those cases are more beneficial actually. For example, Fred Gage's lab actually showed a couple of years ago, it was a stencil paper probably. They used a direct reprogramming approach. At the same time, they also made iPS cells and came down to the desired cell type. And they did not see a lot of the phenotypes that they were seeing during direct reprogramming. And they saw during iPS reprogramming, because you're making a stem cell from somatic cells, you're taking away, you're actually erasing a lot of the epigenetic markers that may or may not.
So probably for those kind of studies and neurodegenerative disorder for direct conversion.
That's probably more.
Can I have one more question?
Yeah, sure.
So those final neurons that you get, they are easily transduced, transfected?
This is
for example, if I want to study
what kind of neurons you're making and what kind of, so they're very easy to infect.
Okay.
Transfection is always a problem in neurons.
Yeah.
Okay, so it's not only
but infect,
neurons.
yes.
The infection is fine.
Okay.
So transfection is always a problem. The method you use toxicity and all. So, but transfection, infection, I can guarantee you, we have infected virus. Yeah.
So you didn't talk about specific ion channels.
Yes, I can talk about it. I can sit down and show you all. But then we have RNA sequencing data, and we have not really looked into individual like KB2.1, for example. We are interested in. Yes, exactly. So we have not really in detail actually looked into what kind of channel. But yes, we have the RNA sequencing data that I did not get time to talk about. It's published already, so we can actually go and look into it, like what kind of sodium channels or potassium channels are expressed in cells.
You looked at what concentration TTX are.
TTX?
Yeah.
You're talking about TTX insensitive sodium channels.
Yeah, right.
TTX blocks most of it. Okay, TTX will block all the action potentials. But is there still sodium current left? I have not tested it yet. I've not looked into it yet.
So you block all action.
Yeah, I don't know if you block resurgent currents, or I actually did not really in detail looked into. So far, we have been really focusing on are the cells functional, are the sodium. We are slowly getting into those questions. Yeah. Okay. I will.
No, but back to the end. So, if you take fibroblasts from two different mutations, they have two different makeup, but they also have slightly different genetic backgrounds.
Exactly.
They have regular.
So there is always subtle differences between different lines. There's some, we don't know what it means. We really don't know what it means. It's always unique. Not only in these rooms, but also like we have ideas as also unique from different lines. There's always subtle differences in their properties. So we have not been.
So it was not that straightforward. Two different individuals were different genetic the direct transformation, right? You get neurons and says, okay, these neurons also.
Yeah, but then somehow you have to also look at the patient brain, the person's brain. Also, like we know that what we see in tissues is actually real transformation that the brain also like, because during reprogramming you are definitely changing several times and changing the. It's also viral infections. That is integrated, so it is not very clean. So this wonderful tool, not very clean. So you kind of have to make a strong case for that by doing multiple lines and different cells, different people, and also somehow have to make sure that these differences really exist in the brain too. So as I mentioned, like there is always subtle differences. We have noticed that there are subtle differences. A lot of times they don't reach significance, but then 10%, 20%
difference of properties that evaluate utility factors.
Well, thank you very much.